Our next speaker is Dr. Stephen Strittmatter. He currently holds the Vincent Coates Professorship of Neurology at Yale and is a founding director of the Yale Cellular Neuroscience, Neurodegeneration, and Repair Interdepartmental Program, and is a founding director of the Yale Memory Disorders Clinic. Now, his work has contributed to defining the molecular basis for brain wiring during development and of neural repair after adult injury. In the aging brain, his laboratory has studied the cell surface molecular interactions occurring in degenerative dementias. He has focused on a missing link in the Alzheimer's disease process, the amyloid beta peptide oligomers that could lead the way to new drug-based treatments, and we certainly need that when it comes to dementia. Please welcome Dr. Strittmatter. The subject is Lost in Thought, Basis for Brain Malfunction in Alzheimer's Disease.
Well, it's a pleasure to be here and to be able to speak with you and to hear all the previous speakers. I guess I need to...
Uh-oh. It's coming. Help's coming.
Lost in thought, lost in computers. All right.
Thank you.
Excellent. So I'm going to focus on Alzheimer's. And I think I wanted to start just by emphasizing the need for progress in Alzheimer's. Of course, everyone in this room has probably been affected from a loved one, somebody you know well with Alzheimer's, and it's a disease that really robs us of what we're about, our intellect, our understanding, what makes us human. And I'm not going to belabor that or emphasize it. I'm going to put up a few slides that emphasize the epidemiology of Alzheimer's and why it's a huge social issue beyond the personal issue that we all experience. So these are some numbers, and numbers get kind of boring, but I think these numbers are so big that we have to actually think about them. So something in this country, some 5 million people have Alzheimer's disease, and an even larger number worldwide, some at least 5 times this number of people, so 25 million. There's new cases of Alzheimer's every 7 seconds, so if I talk too long, more people will have Alzheimer's. I wish I could just stop talking and it would go away. If only it were that easy. It's also an enormous financial burden, one that risks bringing down our whole healthcare system. These are some recent numbers. A conservative estimate is that there's $200 billion spent every year in the U.S. on Alzheimer's care. There's actually numbers from the Alzheimer's Association that are about twice this big, 400 billion. These are huge numbers. It's a massive problem. One of the reasons that this is true, maybe it's unfortunate, but this has been alluded to earlier, we're getting better, perhaps, at taking care of other illnesses, which makes Alzheimer's a bigger problem because we're not good at treating it, at fixing it. And this is just a little schematic showing as a cause of death, Alzheimer's is increasing over the last decade, whereas these other conditions we're actually making some progress on. And coupled with this kind of trend and the relative increase in the elderly population, and of course Alzheimer's is a disease of the elderly, this number, which is already huge, is predicted to triple by the year 2050 in this country. So there's huge need for progress in Alzheimer's disease, and I think we all know that. These are just some numbers which bring it home. So what do we do about Alzheimer's today? What is therapy for Alzheimer's? And unfortunately, we have very few weapons in the fight against Alzheimer's today that we can use in the clinical situation. There are some approved medications, but they really provide symptomatic benefit. While you're on them, it helps a bit, but it really doesn't change the course of the disease. So we really don't have this, a disease modifying, something that slows down or reverses the disease. And it's been alluded to earlier that it's been sort of a disappointing last several years or decade of attempts to develop this kind of disease-modifying therapy have failed to meet their ends, and we still don't have this kind of desperately needed therapy. So there have been responses to this, and Dr. Hodis alluded to this earlier. One approach to this idea, why these things have failed, is that they've been used too late. So when the disease has already damaged the brain to a point where it's hard to pull things back. And so now there are attempts to treat earlier, and trials that the NIA is supporting are trying to make that happen and see if therapies that we have will work at an earlier stage. I think a key thing is that we need to find new drugs, new targets, and test them and develop new therapies. And I'm going to emphasize the point that this depends on molecular understanding and tell you about some of our experience. And I just want to make one point here. Again, already alluded to, there is this national plan to address Alzheimer's disease, and this is important progress, and it's expanding our effort to make a difference, to discover these therapies. But I would point out, even if under the best sort of budgetary scenarios, we may be able to, with this plan in place, get to a research effort that's still something like 1/1000th of what we already spend on Alzheimer's care. So I would emphasize that more effort would be in all of our interests. It's the only way we're going to get to these disease-modifying therapies. So now I'm going to dive into Alzheimer's and tell you about what we do know about it, approaches that we've taken to it, and where we might be able to go forward. So this idea of the amyloid hypothesis of Alzheimer's disease has been around now for a number of years. There are chinks in its armor. It's still probably the most prevalent idea about how Alzheimer's comes about. Amyloid plaques are something that probably most people have heard of, this sticky substance that deposits in the brain. When the brain of someone who's died of Alzheimer's is studied in the microscope, there are these plaques, and I'll show you a picture of these in a second. The major composition of these plaques is this single peptide called amyloid beta. And this observation decades ago is really what led to the idea that it may be causative in the disease process. This single peptide amyloid beta would somehow trigger the disease. There's also evidence from, as mentioned earlier, the rare families where a single gene mutation can cause Alzheimer's. That's not what most Alzheimer's is, but a few families carry a gene, one of several genes, where a single mutation can introduce the disease. And those mutations, by and large, are in the pathways that produce A beta itself. And more recently, sort of over the last 5 or so years, there's been a large consortium effort to study the progression of Alzheimer's disease, again, funded in part by the National Institutes of Health. And this kind of study of the progression into Alzheimer's, so this is progression over time from a schematic here, if one tracks patients as they develop dementia, go from cognitively normal to mild cognitive impairment to dementia, really the first thing that's detected, whether by PET scanning or by CSF, spinal tap studies, are changes in A beta. So it's the first event. It precedes, perhaps by decade, the eventual progression of dementia. So there may be later things that affect tau pathology, loss of brain tissue, and eventual dysfunction of the brain. But this is another reason to think that a trigger, very far upstream, is this amyloid beta peptide. So how does this actually affect the brain? So this amyloid beta peptide that I've been talking about is what deposits in these plaques shown here in red. It comes from a gene called amyloid precursor protein, gets chopped up, and actually most of the things that have been tried in the clinic recently that haven't succeeded have tried to alter this conversion from APP to A beta or try to pull this A beta out of the brain. So this material accumulates, but there's recently quite a bit of data that there's several forms of this A-beta peptide. When it's first made, it's simply a monomer and it floats around, and we actually all have plenty of it. Actually no different than in Alzheimer's patients, really. Then it can form small clumps called oligomers, and I'm going to show you a picture of those in a second, or it can go on to develop these huge plaques. And the data suggests that it's actually these oligomers that make the brain sick. It's not these plaques, but it's the oligomers that are unique to Alzheimer's disease. Once they interact with neurons, they can impair synaptic function. This eventually leads to loss of neurons, these tangles, neurofibrillary tangles in the brain, and it's this whole constellation of events which actually produces the dementia. Now, a key part of this, and something that we've focused on, is the idea that this material is outside the nerve cell, and this sort of derangement of brain function is happening inside of nerve cells. So how does this toxic material trigger this bad stuff in a neuron? I'm going to give you a little better picture of that, and this is actually from the NIA website. This is meant to be a cell membrane here. This APP protein that I was telling you about releases A-beta, and eventually it piles up in these plaques. That just gives you some sense of scale. And over here is the normal brain. Each one of these big blobs is a nerve cell. They send out all these processes. You can think of them as wires. They conduct electricity. They have synaptic connections, it's called, where one cell touches another. And an electrical impulse that may travel down an axon releases a chemical and is transmitted to an electrical impulse in the second cell. And what seems to be happening, the evidence is that this A-beta oligomer can cause a disruption, a dysfunction in these synaptic connections. So how does this happen? What is this oligomer? How does it interact with the neuron? That's what I'm going to talk about. Now, these are actually some pictures of A-beta itself in an electron microscope that we took. The single peptide doesn't have any structure. Once it makes these oligomers, you can see these dots. And then when it makes this fibril that piles up in plaques, it looks completely different. Now, we don't really know in a molecular sense how this is organized, but these are some models that have been produced in part with experimental evidence from David Eisenberg's lab at UCLA. And in this state here, there's really quite a different structure. The two A-beta molecules, one's pointed one way and one's pointed another, and they're twisted in what's called a cylindrin state. The key point is that this blue stuff is very different than this yellow stuff. And it's the blue stuff which may be most toxic to the neurons. So we decided to look into this. How does this blue cylindrin toxic stuff interact with neurons? So what's shown here are nerve cells in culture. And what we did was take this A-beta oligomer and tag it in a way that we could see where it binds to the neurons. And you can see that it binds to neurons. And actually, in the background here, there's other cells that it doesn't bind to. It binds only at a certain stage of development when they start making synapses. The monomers don't bind. So all of these things suggested to us that this oligomer had a specific place on the neuron that it would stick, a lot like a lock and a key, a very specific site. And what we decided to do was to try to figure out where this A-beta oligomer was binding on the neuron. What's the molecule that's responsible for this, and therefore allowing the downstream bad effects of the oligomer on neuronal function. Now, this is a little bit of molecular biology. I'm just going to say a few words about. We used a method called expression cloning. So the basic idea here is we didn't know what this molecule was, but we figured we'd check every gene that there is expressed in humans. And the way we did this was to take a cell that doesn't bind these A-beta oligomers, and then one at a time add the genes from the brain into different cells and ask, is there any brain gene which will create these binding sites similar to what we see on the neuron? And there was one that did, and that's the result is shown here. So here, these are non-neuronal cells that don't bind A-beta, so they're not dark. And what we did here was introduce this one gene, which I'm going to tell you about, called prion protein. And when this protein is in these particular cells, now they'll bind the A beta oligomer. In fact, this binding event looks just like what you see on a mature neuron that has synapses. So this single prion protein was the only gene out of some 200,000 that we screened through that allowed this kind of binding. And it suggested to us that this would be important in the function. So let me tell you just briefly what cellular prion protein is. So you may have heard of this protein before because it can misfold and become infectious in what's called mad cow disease. And in fact, there's evidence that this protein is involved in the degeneration that happens in this other brain disease. So we figured that's what's going on in Alzheimer's disease. So we wanted to test whether it's really important for the synaptic dysfunction. So we do that in a piece of hippocampus. So this is a part of the brain that we put in a dish and we record electricity. And that's what's shown here. We stimulate one of those axons that you saw before, and then we record the synaptic connection. And if you do this in a healthy slice, you can record this electrical potential. And then if you stimulate it a bunch and record the potential, it's greater. This is, in essence, sort of a memory that happens in this chunk of brain tissue. And what had been shown before by a number of labs was that if you do this in the presence of this A beta oligomer, this potential is suppressed. So they can't remember the fact they got jolted here. And if you do this entire experiment, this effect of the A beta oligomer in a piece of brain tissue that genetically doesn't have prion protein, now there's no effect. So the slices are protected from the bad effect. They require prion protein in order to have this bad effect. Another thing that happens later is that synapses are actually lost. So this is zoomed in on one of these pieces of a neuron in culture, and you can see each of these little synapses. And if you watch them over several hours, you can see that some of them in the presence of A beta go away. They shrivel up. They're lost. This is what we worry happens in the Alzheimer's brain. And so we did this kind of experiment in neurons that don't have prion protein, and these synapses aren't lost. Instead of losing 10 or 15% of the synapses in this time period, without prion protein, the neurons don't lose their synapses. And then we wanted to see if it was important really for something that's a little bit more like dementia. Can we test memory? Now, there's not any perfect model of Alzheimer's disease that we can test in the laboratory, but as mentioned before, there are these cases of Alzheimer's disease where single genes can cause the effect in humans. And so these have been transferred into mice, and the mice get plaques. This is a piece of brain with a bunch of those plaques. And the mice get memory loss. And this is just a picture of how we test memory. So the mice go into a big tank of water, and somewhere in the tank of water there's a platform where they can climb out of the water. And after they've gone in the tanks a bunch of times, they learn that the platform is here. And so a healthy mouse, when it's put in this thing, will go directly here and swim around to get out of the water. These are the Alzheimer's mice that have these two genes from human Alzheimer's cases. They don't know where to go. They wander all over. If the mice have these Alzheimer's genes and have plaques, but they don't have this prion protein, now their memory is over here. It's improved. So getting rid of this prion protein might actually benefit the ill effects of A beta oligomer. And in fact, a study at NYU showed with an antibody to prion protein that these Alzheimer's mice were corrected in their memory. And there's NIH support now at Yale to look for drugs that will block this prion protein and achieve this kind of genetic effect. And also the drug company AstraZeneca is working in that direction with antibodies. So I told you about this A beta oligomer binding to prion protein. In studies that I'm not going to go into, we've actually mapped out a couple of the molecular steps in the neuron right after this happens. And one of these is, after prion protein, is this molecule called mGluR5, and then a second one is this molecule called FIN. And the reason I mention these as being important, these second steps, is that there are known compounds that interact with each of them. So this molecule MTEP blocks this blue protein, mGluR5, and if this is given to these same Alzheimer's mice, it has a beneficial effect. So this is, what's scored here is actually multiple swims as the animals are learning. So wild-type mice quickly learn where that hidden platform was. That is, they go to the small circle in that tank I showed you before. The Alzheimer's mice, when they're aged, they don't do well here. So they learn slowly, and additionally to having bad memory later. But if you treat these Alzheimer's mice with this drug, now they learn just as well as healthy mice do. Their time to get to that platform drops right down. And so this is something that we can hope to think about studying in humans. And there are actually, here at Yale, it's been developed ligands, PET ligands, that can image this particular blue molecule in a human brain using positron emission tomography. And the third step in this process is this molecule fin kinase. It's also critical for this whole pathway. And in fact, here again, if you see the synapse loss in a neuron that doesn't have fin, it doesn't happen. So this fin kinase is actually a member of a 4 or 5 kinase family called the SRC family. It is a tie to this phospho-tau protein and the tangles of Alzheimer's disease. So it really knits the two major pathologies of fibrillary tangles and A-beta plaques together. But importantly, there's a drug that targets this kinase. It was originally developed for cancer. It doesn't work too well in oncology, but it's safe. And there's actually now a Yale-initiated trial to test this in Alzheimer's disease. And this has been funded, again, by the National Institutes of Health. So I'm going to conclude here and make a few points. I told you about these 3 molecules, potential new targets to approach the Alzheimer's problem. This is certainly, there's many approaches to the problem. And I think we need to pursue multiple lines of evidence so we can actually get one of these disease-modifying therapies. Tau pathology downstream here, not only is it downstream of this pathway, once it's triggered, it can actually maintain itself and spread. And this is an active area of investigation to try to block the ongoing tau pathology. And as Dr. Hodes mentioned, the immune system plays a big role in regulating the responses to A-beta, to tau, and the entire synaptic dysfunction that's in the brain. There's also a lot of evidence that Alzheimer's disease interacts in a sort of multi-disease fashion with both diabetes and vascular disease. Those are important additional places for interaction. And we're also making a lot of progress in other forms of dementia which teach us something about Alzheimer's disease, the 2 major ones being Lewy body dementia and frontotemporal dementia. And in conclusion, I think we need to know a lot more about the molecular mechanisms so that we can get to a disease-modifying therapy to stop this burgeoning, this tsunami of Alzheimer's disease that threatens to overwhelm our healthcare system, and on a personal level is devastating to those whose intellect is robbed. And I'm just going to stop there and mention the people in my lab who did this work, and most of this has been supported by various organizations in the NIH. Thanks.
Thank you, Dr. Strittmatter.